So calreticulin is particularly amenable to targeting with an antibody for a number of reasons. So we know that when calreticulin mutations occur in myeloproliferative neoplasms, they cause the calreticulin protein to be changed, and in particular, what we call the C-terminus, or the end of the protein, is completely altered and has a new sequence, and that's only present in the MPN cells. So the normal wild-type cells do not have this new C-terminus, just the mutated cells. And that's a specific sequence that's unique to the MPN cells, so that makes it a very attractive therapeutic target. The second part is that we know that the mechanism by which calreticulin mutations cause MPN is that the mutant protein binds a receptor called MPL that's normally expressed on the cell surface. And so what happens then is that these two proteins form a complex that is expressed on the cell surface. And this, again, highlights why it's attractive to think about an antibody, because an antibody binds outside the cell, so you need something that's amenable to targeting on the cell surface. So mutant calreticulin binds MPL. This complex of the two proteins goes to the cell surface, and it has this specific sequence that's only present in the mutant protein, not in the normal protein. And so that makes it particularly exciting and attractive as a therapeutic target, because if you had an antibody, it would be specific to the MPN cells, and the normal cells would not be targeted with the antibody since they're not expressing the mutated protein. And so what I presented at the MPN Congress is exciting developments that are in what we call the preclinical stage of development. So this is everything up to the point of treating a human being in a clinical trial. And there are now 3 independent studies, one from Japan, one from Austria, and one from Australia, where 3 groups have developed antibodies against the mutant calreticulin protein and have shown preliminary data in preclinical models, so either using mice or using cell lines that you culture in the laboratory, or taking patient samples and treating them with the antibody in the laboratory, being able to show that these antibodies do seem to bind to the mutated calreticulin cells and inhibit their growth. And so that's very exciting if that's able to be translated into the clinic, because this would be a novel or new treatment that would be very specific for the cells that have the calreticulin mutation.